Hello, everyone. Welcome to this program. We're going to change things a little bit here so that everyone can see the screen. What we'll do today is we'll give you an overview of the program. I will pass this on to Sparsh momentarily, but to give you an introduction, Bioinformatics for Infectious Diseases is a program designed to introduce you to the ways that bioinformatics is used in the study for infectious diseases coming from different types of pathogens. And what we'll do is we'll introduce you to curated examples of how you can analyze data, where to find such data, and how to make conclusions. Okay, so what I'll do in my part of the presentation is I will review some of the introductory material about the program. Then I will pass it on to Dr. Raghavendran, who is going to review some of the practical things of how you can get started with some of the types of data that is usually used for these kinds of studies. So Bioinformatics for Infectious Diseases is a program that is designed for biologists, clinicians, students that are interested in virology, microbiology, and immunology that would like to learn how to apply bioinformatics to different types of big data for research and applications about infectious diseases, to design novel diagnostic approaches, to understand how antiviral and antimicrobial drugs are being designed, how vaccines are relying on data and bioinformatics to address new and maybe potentially in the future upcoming disease outbreaks, and also how to understand what are some of the important challenges and questions that remain to be answered, and how bioinformatics can help us address those as well. So what I'll do is I'll first explain how some of the questions about infectious diseases are related to bioinformatics. If we can think about this program as just a series of questions that one can ask and use bioinformatics to find potential answers. One question might be, what is a pathogen? What differentiates pathogens from non-pathogens? What differentiates, for example, bacteria that coexist together with humans, plants, and other hosts from those that cause disease? How can that help us understand the evolution or the conditions of competition that cause these microorganisms to evolve, share recipes, kind of record what works and how to adapt the tools that they have to overcome different types of challenges? Another question can be, where do diseases come from? How come is it that some diseases persist and some diseases have these outbreaks that are acute, that have a beginning, that have an end? Where do they come from? How do we understand the origins of diseases like coronavirus or Ebola or other diseases that cause these endemic or pandemic outbreaks? How can we understand this in the context of interaction between different hosts, for example, different species, humans, their pets, the different cattle that we grow? How can that be a source of novel pathogens? What is the actual disease that they cause, right? How can we describe the reaction of the host to the pathogen as a condition that we can diagnose? So what makes that as a disease? And we can think of all of these microorganisms as essentially parasites that leverage some of the resources that the hosts have accumulated, leverage their mechanisms of production of vital proteins to be able to replicate themselves, and how they, as a result of hijacking some of these resources, can also cause toxicity and disruption of the host function. We'll also talk about how diseases spread, because what characterizes an infectious disease or a communicable disease and sets it apart from other types of diseases, which are commonly known as chronic diseases like cancer, for example, or hypertension, is the fact that they're caused by a pathogen, and that pathogen is a transmittable pathogen. You can actually share it between individuals. And so when we think of that transmission and the pattern of transmission, we can also use bioinformatics to understand the population scale. How does the population react to a disease, for example, herd immunity, or how does the population become susceptible, for example, having similar characteristics within a particular group or a population, how can that make them more susceptible to particular types of diseases? And we'll also talk about the balance of replication, how while a pathogen causes acute disease oftentimes, one of its objectives is to continue and replicate and find a balance where it can continue to replicate and spread between many individuals, causing them to potentially have mild condition that does not prevent it from continuing to replicate. Then we'll talk about how to treat or prevent disease, and we'll talk about how novel advances are examples of bioinformatics in part because understanding the particular situations where vaccines can be more adapted to the specifics of the pathogen, and maybe novel antivirals could be designed based on our understanding of the interaction between the host and the pathogen, can lead to novel ways to treat disease. But we'll also talk about how early diagnostics, or understanding specifically what pathogen and what strain of a pathogen, or maybe a variant of a pathogen, is causing a disease, is critical for the future of these kinds of interventions. Because as we will see in the last portion, what remains to be done, we'll see how the same principles of evolution can help us understand how the diseases that are caused by pathogens are only preventable if we can develop systems of early detection and understanding of the evolution that they undergo, because we can identify a precise variant and we can treat that precise variant, but we don't always know how to anticipate novel variants. And to anticipate novel variants and design therapeutics that can potentially co-evolve with these pathogens, there are new strategies and novel understanding about how these types of discoveries can really help us anticipate and prevent future outbreaks. So in the context of this particular online program, what we have prepared on our side are a number of examples, or let's call them case studies. Case studies where we can take curated data sets and start designing bioinformatics projects where we have a question, we have a data set, and then we have a path to answer that question from that data set, or methods of analysis of data that could inform some of our understanding of the answers to these questions. So what are some of these case studies and data sets? One of them is going to be about the Ebola outbreak in 2014 to 2016. Now, what's interesting about this outbreak is that it is one of the first examples where large-scale genomic sequencing was employed almost in real time to track an epidemic, to understand how the epidemic is spreading in a particular region, and then use this understanding to design a vaccine and an antiviral drug. Another example that we're going to look at is going to speak about the spread of COVID-19. And when we think about COVID-19, we obviously hear so much about it in the news, but what is exceptionally, I think, kind of for bioinformaticians is interesting in this pandemic specifically, is that the amount of data and the detail of data is now very rich and very precise, being updated almost in real time from many different regions. So really the scale of the data collection here is unheard of in any other kind of disease. Not only that, this particular pandemic has popularized some of the terms that we rely on as bioinformaticians to understand how genomes of particular pathogens or how molecular biology and kind of the background of how these pathogens cause disease are really becoming understood by even the general public. So today, everyone understands what a variant or a mutation is. Everyone appreciates the ability to study some genomic pattern and try to make inference that has a practical application. We don't have to explain the need to understand and maybe sequence some of these pathogens, because now everyone is in tune with this scientific endeavor to try to really understand how this pathogen is evolving, right? We don't need to explain that it is evolving, right? Everybody knows that. So really, we're going to take a look at some of those data sets in this as well. Another example that we will look at is the development of vaccines to address the kind of chronic or endemic Plasmodium falciparum spread in Africa and in South Asia. We'll take a look at how genomic surveillance and understanding of both resistance to some of the known drugs that are used to treat Plasmodium, as well as the efforts to design an effective vaccine that turns out to be a non-trivial effort that requires a deep understanding of genomic variability across different regions and the understanding of how that genomic variability can be used or leveraged to really find an effective way to interfere. We'll also look at how viruses are not only the result of these kinds of acute outbreaks of infectious diseases, but we'll also see how there's a greater appreciation of the role that viruses play in other types of diseases. For example, in neurodegenerative diseases like multiple sclerosis, where maybe an exciting idea resulting from this observation is that if we treat the virus, if we find a way to treat the virus, the viral infection, we don't have to deal with the symptoms, with the consequences that are the outcome of this persistent infection leading to these long-term deterioration of the cognitive function. We'll also take a look at how these types of observations are relevant in diseases like cancer, where Epstein-Barr virus and others have been found to play a major role in the onset and maybe development of these cancers. So I think really infectious diseases now is going from some of these prominent case studies that we're going to take a look at, like Ebola, malaria, and COVID, and even extend to some of the non-typically considered as infectious diseases, but chronic diseases that potentially have a connection to viral pathogens and others. We'll also look at some of the challenges that still remain to be addressed, even though we think of treatments like antibiotics, for example, as an established way to treat disease that have been very successful over the past several decades. These kinds of interventions are new. Our ability to design vaccines and adapt them to particular pathogens is something that happened in the past maybe decade. Our ability to leverage the antibiotics to treat these bacterial diseases is also just a few decades old. And as a result of widespread use of these types of interventions, we're also seeing the reverse reaction, right? Where we are seeing how these types of interventions can become weaker over time as a result of evolution and adaptation. We see emergence of resistance, and drug-resistant tuberculosis, for example, is a major risk. We can see novel pathogens, bacterial pathogens that are emerging in hospitals that are not possible to treat with antibiotics. We're also seeing novel variants of these viral diseases that also do not respond well to vaccines that were designed. So really, these are some of the challenges that continue to emerge in these kinds of effective ways to leverage bioinformatics. And we'll talk about where we can find some data sets and what are some of the methods of analysis that we can leverage to try and answer some of these questions and challenges. Now, where can you find some of these? We have designed a portal where our team has curated project examples and tutorials that you can leverage as an independent learner, as someone who just wants to obtain some bioinformatics skills, or as someone that would like to apply some of the bioinformatics skills to a particular research question. And so those are some of the things that you will find on the portal, and I will explain today how you can leverage this portal more effectively in the context of this online training portal that is going to provide you with live sessions, as well as some of these online resources that you can leverage to really master some of these skills and practice them on particular examples. So to talk about this, I'll speak about where you can find a particular resource that you might be interested in, how you can find different categories or even collections of different courses, and we'll see how to get started. Now, beyond this, I want to mention that what you will hear in this program is going to be based on tutorials that were prepared in collaboration with a number of experts in this domain. So really what we did is we worked with a number of researchers that are actively investigating all of these topics from the perspective of pure bioinformatics or an algorithmic perspective, how to design methods to analyze such data effectively. We've worked with researchers that specialize in some of these pathogens and have been performing research in the area of malaria or coronaviruses for a long time, and also people that apply the bioinformatics to connect the biological questions with the specific data sets that are available and are being extended and populated with these new viral and bacterial genomes, or even parasite genomes in the case of malaria, to try and answer these questions comprehensively. So we'll leverage their expertise and we'll present to you some of the findings and questions and solutions that they have identified. So what is this program all about? This program, Bioinformatics for Infectious Diseases, is going to help you understand pathogens and the diseases that they cause, where they come from, what are the processes that cause the different types of changes in their genomes that we can observe, how can they cause infection, and how can that infection cause disease. We'll also talk about the specific ways to analyze such data. A lot of this data is going to be focused on the understanding of genomic variability and how to identify specific genomic variants and characterize them in terms of their impact on the pathogen itself, on the interaction between the pathogen and the host, and potentially on the intervention in that interaction. We'll also talk about how you can apply some of these principles to study a variety of different curated data sets, identify the questions that are important for these data sets, and then use the data sets and the methods that we describe to find answers to these important questions. And again, you will have access to some resources that will make your life easier if you're just getting started, but also for those of you that are already more advanced, we will help you connect the dots and really apply these methods in an effective way. Now, the program is going to help you by leveraging two primary resources that I want to introduce briefly before we begin. The first of them is going to be our big data analysis platform. Some of these data sets are going to be exceptionally large. We're going to leverage hundreds, if not thousands, of genomes at a specific time. We're going to look at complex data like RNA sequencing, even single-cell RNA sequencing. And so to facilitate some of the educational aspects of our interaction, we're going to rely on this platform that I'm going to demonstrate in just a few moments. The other part is, of course, our Omics logic portal that I already briefly mentioned, but I want to highlight the significance of this online resource because it provides a collection of elements that are going to be useful as you start in this particular program and continue after that, so that you can understand the high-level overview in a short program like we have today, but then continue by leveraging some of the specific technical tutorials to really expand and practice and master these skills so that you are independent and confident in the way that you apply them. So before we begin, what I would like everyone to do is to log in to this portal. So let's go to learn.omicslogic.com. This is a portal where you can register for free. So anyone, even if you're not going to participate in this particular program, you can register. And I'm putting this link here so that all of you who have not registered as of now can take a moment to register. Now I'm going to demonstrate, and please follow along if you have not registered already. So I'm going to log out. The first thing that you will see right here in the menu is this login screen. Okay, so if you press on the login, you can either log in with your email and password. You can create an account. Again, this is a free account. You put in your name, email, and think of a password. Or you can also use different types of social media plugins. So after you have created that, I'm just going to use my own password to log in. And the first thing that you will see if you are registering for the first time is actually the pricing page. But if you go from the pricing page, you should see right here on the very top your email address. And so that's what we want to do, is we want to click on this email address right here. So this email address is your profile. Okay, so please let me know once you have your profile in front of you. I'm going to walk you through some of the things that you're going to be seeing. So just let me know in the chat by saying yes, putting number 1 or something in the chat so that I know that you're with me on your profile. All right, excellent. Thank you, Brian. Eileen. Okay, I'm going to give another 30 seconds for those of you that are still creating your profile. Please let me know once you're done. All right, thank you, Priyanka. Sarah. Excellent. Okay, Anna. All right. So this profile is really designed to kind of connect all of the different resources. Naveen, the creation of the profile itself is free. So really, shouldn't be any worry about cost right now, right? The profile is free. You'll have access to a lot of free resources. If you have questions about pricing later on, please reach out to our marketing team. But right now, I'm just showing you a free resource that you can already leverage from day one. So in this profile, you'll see how the different resources are connected to your learning journey. And we're kind of emphasizing this section because really this is where you can track your progress. You can go back to where you were to pick up where you started. You can always find your certification here, etc. So all of this material is going to be here. What is the expectation of this particular program? This particular program will help you identify the specific topics that you need to master, try to apply them to different projects, and then use the annotation of our tracking of your progress to earn a certificate. And so this certificate is going to be the validation of your kind of understanding of bioinformatics in infectious diseases, number 1. But number 2, it will also provide you with an opportunity to develop an independent research project. So one of our primary objectives in these kinds of training programs is to support research that is coming from a bioinformatics perspective. How to apply bioinformatics to research, that is what we're really going to address in this training program. Now, this training program will have a dedicated program page. On that page, you will find all of the sessions that will be recorded. You will also find a schedule of how to go to the right kind of a topic at the right time, and how to connect to the live sessions that we have today, like the ones that we have. Now, beyond that, you will also find all of the resources. Now, we understand that many of you are coming from different backgrounds. You might be already an expert in bioinformatics, and you would like to understand how specifically bioinformatics applies to infectious diseases. Or you might be a complete novice, and you really don't know a lot of the topics that we are discussing. Maybe you're not familiar with the terminology, for example. So that's why we include in this particular program also the introductory coursework, like Bytes and Molecules, Introduction to Bioinformatics, and others, where you can start with the very basics and develop your understanding of the different terms that you will find useful as we go through the different case studies. As I mentioned, we will have some live sessions like today, but we will also have recorded sessions. And so you will find those in the schedule, and you will finally be able to interact with our community by asking them questions about the specific topics that we are discussing and getting maybe technical answers to overcome your challenges. So if you ever get stuck, and if you ever have maybe a frustration like always happens in these online courses, I don't understand, I can't do what I'm being told to do, you can always reach out to us. So our team is going to be there to support your journey, and you're going to be able to then go through the specific tutorials that we recommend to learn about that. Now, if you don't see any courses, that is because you are likely just signing up. So if you go to this page, the course tab has the courses that you've already started, okay? If you want to find a course, you have to go to the top menu, and this is our library of resources, and find the relevant course. For example, if I would like to learn about COVID, I will find SARS-CoV-2 genomics as an introductory project right here. So once you start this, and you start by clicking on the lesson, and now you've started this project, you can learn through and then click on next, and eventually that will appear on your profile as well. So Christine, hopefully that answers your question right there. Okay, so finally, before we go on, I want to recommend you to connect to our community as well. So what this program represents is a community effort. Not only are we going to have these kinds of sessions and interact with you, try to help you, try to explain some of these topics in an accessible, approachable way, and really distill these complex topics to some simple exercises that anyone can perform. We're also there to connect a community of learners to opportunities. And those opportunities might be professional opportunities to develop your career in bioinformatics, also opportunities to connect with potential research collaborators. So we're going to introduce you to some of those groups. And today I'm just going to share here in the chat 2 of our groups that you can connect to. One of them is on LinkedIn, and another one is on Facebook. For those of you that are on either one of those platforms, we welcome you to connect with us personally, but also to connect with the broader community that we serve. We have over, I think, 1,500 people on LinkedIn, over 10,000 people on Facebook. So you can really leverage this community to not only learn about bioinformatics for infectious diseases, but then also to continue and apply bioinformatics to other domains and to advance your career or find opportunities for research. So today's session is going to be dedicated to next generation sequencing. One of the technologies that has really advanced our understanding and ability to treat and diagnose diseases like infectious diseases is our ability to sequence genomes and transcriptomes and understand what's going on at the molecular level. So when we think about next generation sequencing and the way it has become popular and maybe widely used as a technique, this has happened in the course of the past couple of decades. And today, with decreasing costs of sequencing, it is being used in remote areas where some of these diseases are originating, where some of these diseases are being changed and transformed, but also in most kind of technologically advanced places, it is becoming a more and more reliable and useful technology to understand infectious diseases. So to learn more about this technique, I am going to invite Dr. Raghav to speak about how next generation sequencing works, what it contains, what kind of information it contains, and how you can potentially start learning to use it to actually apply it in the context of infectious diseases. So Raghav?
Yeah, hello everybody. Let me find ways to share my screen. Okay.
Are you sharing your screen?
Yeah, it's my PowerPoint is not responding. Just a second. I'll quit and rejoin.
Okay, while he's rejoining, I will get started with that presentation. And then once he's back, he will take over. So let me share my screen again. 1 second. Okay, so one of the things that we know today is that the viruses, bacteria, parasites, and other pathogens surround us and coexist with hosts like humans. And one of the things that we would like to understand is what differentiates specific pathogenic microorganisms from non-pathogenic microorganisms. So this leads us to this concept of a virome. Viruses are obviously some of the most clearly understood pathogens. They actually are not characterized as living microorganisms, but they only live in a host. They can only replicate within a host. And when we think about viruses, we can think about the virome, or the collection of all the viruses that are present in a specific environmental sample. Environment here can be the human gut, the human lungs, it could be the water that we drink, or it could be anything around us. And so virome or microbiome are these collections of microorganisms that we can sample and clearly identify. Now, within the virome or the microbiome, we can start characterizing the specific individual microorganisms. And one of the important things is that because of the next generation sequencing technology, we now have a variety of viruses, bacteria, parasites that are characterized at the genomic level. Now, the genomic revolution that originally started because we wanted to understand the human genome has in parallel led to an explosion of the number of viral, bacterial, and other types of microorganism genomes being sequenced and studied. And here you can see just a glimpse of how much information is available. And you can see here that the most studied viruses, for example, are the viruses that are commonly found to be pathogenic to humans. Now, obviously, we can kind of understand the logic behind this. For example, just if we look at the scale at which COVID-19 is being sequenced, we can see that the major outbreaks of infectious diseases like SARS-1, Ebola, Zika, MERS, and now COVID-19 have been these spikes in sequencing. Why do we want to sequence these genomes, and why particularly do we want to sequence pathogenic genomes? Well, one of the things that we want to try to understand when we're sequencing these pathogens is the specific recipe that these pathogens employ to be effective at what they do, which is to replicate, evade our immune response, and ultimately adapt to the different types of responses that typically work, but in their cases, they're not working, to overcome and continue and spread within the population. So these pathogens that we are going to study in this program are going to be studied using next generation sequencing to understand their genome. And then we're going to apply the same technology to understand the response. How does the human immune system respond to these different pathogens? How does it vary across different types of disease manifestations? And how can we characterize those interventions that we can design based on this interaction that we understand because of next generation sequencing data? So really the critical thing here is to understand this technology. How exactly does it work? But also, how can we analyze such data, and how can we translate the analysis to biological insights that are actionable, that are informative and actionable? So what is next generation sequencing? And now we're going to start with thinking about next generation sequencing in the context of pathogens. How can we use next generation sequencing to understand the genomic structure of viruses and bacteria? And how can we learn to identify important variation that we think leads to their manifestation of disease? How do they cause disease? Metagenomic and viral sequencing can be of several different types. Specifically, we can look at targeted regions that we already know. For example, ribosomal RNA is one way to look at only bacteria, because bacteria have ribosomal RNA that is different in structure from, for example, human ribosomal RNA. And so that's how we can amplify a particular type of a sequence to identify only bacterial DNA or RNA. Another example for targeted sequencing is what we hear for SARS-CoV-2. When we only want to look at for example, variants of concern that affect the spike protein. And so then we know how to amplify only the region that contains the sequence for the spike protein. So we can sequence particular regions by amplifying them, and that's called targeted sequencing. Another type of sequencing is total DNA or RNA. When we're not looking for any particular region, but we're saying, let us just extract all of the genomic material from a particular sample and sequence everything. And then finally, you can see here, amplicon environmental. In the same example, when we have a particular type, so one type targeted would be, let's target the spike gene. Another one, let's do a type of a sequence, and we'll understand the composition of a particular sample. Now, these are very different approaches, but they rely on the same technology. So to help you learn in detail, we recommend these three particular articles that you can read through on your own, and we'll share at the end of today's session the links to these articles that will help you understand how these types of sequencing approaches are helping us understand the presence and characteristics of different pathogens. Now, let's talk about sequencing specifically in the context of viral communities and bacterial communities, right? So microorganisms, again, pathogens. So a lot of times, one of the challenges with metagenomics, when we have an environmental sample that contains a lot of different present organisms, we would like to understand how all of these different organisms are characterizing the environmental sample. Now, in bacterial examples, there's always a challenge, because with bacteria, we're not able to isolate and grow individual bacteria. Only 1% of bacterial isolates could be grown in the lab. So one of the questions would be, well, how do these bacterial communities exist in real life, in the real world? And to do that, we have the approach of metagenomics. With viruses, it's the same thing. Not only do we have viruses interacting with other viruses, these viruses also interact with other bacteria. In fact, the majority of viruses are bacterial viruses, viruses that only infect bacterial cells. So when we think of a particular virus that is causing a disease, we would like to understand the community or the environment where that virus came from. And in many cases, we would like to characterize that particular one pathogen, how that pathogen is different from the others that it lives together with, or that interacts with others like them, and then understand how the sequence of that particular virus can be a piece of information that we can use. how can we use that piece of information? In viral outbreaks, we would like to take a clinical specimen, for example, a human blood sample, and we would like to find all of the viruses that are present in that clinical sample. For example, if we have a temperature, if we have vomiting, if we have cough, right? We want to understand what is the particular pathogen causing the manifestation of that disease. And to do that, what we can do is we can sequence everything that is in that sample that includes the host genome and maybe genomic material, as well as anything else that is present there. Sometimes we already know. So if we have a particular virus that we think is causing the infection, right, we can design a primer, specifically amplify those regions that we know should be present based on the virus strain that we think is present in this particular patient, and then try to understand, is that the same strain that we expect it to be, or is it somehow different from the reference that we have? Now, in many cases, we also have multiple causes of a condition, right? And so what's important at the first stage is to try to understand what all is present in a particular sample. And we can do that because we have different types of data where we can store the resources about potential organisms that we can find. For example, we have a database of microorganisms or a database of viral genomes, and we can now compare and contrast what we find in a sample with what we will find in a particular sample that we have, or what is known to be in a database. Now, to understand how these different techniques are going to be useful in the context of next generation sequencing, we have to understand the types of files that are used to store and retrieve such information. So I think Raghav now is ready to take over. I think he's got his presentation working as well, right, Raghav?
Yes, I got it. Sorry for the interruption, but I just had to restart my system, something. Yeah, my Zoom and PPT never spoke with each other. Okay.
Please go ahead.
Yes, thank you very much. Okay. So yeah, just confirm me that stop my video.
Yes, we can see your screen.
Yeah, you can see my screen. Yes, okay, that's fine. That's great. But I think you cannot see my video because, yeah, just a second. Let me. Okay, now I can. Yes, great. Perfect. Okay, let's. So we already learned about why and how NGS is going to help us characterize different whole genome sequences of these infectious diseases, and why, in the context of why it is going to be very important in perform an analysis like next-gen sequencing analysis, sequencing methodology to identify or to sequence the whole genome of pathogens. So let's go through some of the important concepts or some of the important outlines of how next-gen sequencing data is generated and what are the different file forms that are associated with the next-gen sequencing data, and how we can use the Taubot Bioinfo platform to run a pipeline for this sequencing data analysis. So yeah, so this is very important because it is related to our ability to compare the DNA code of any given system, in this case like a virus or even a bacteria, to a reference sequence of the same system. Like if you want to identify a new strain in the, that is circulating around during this pandemic, then we sequence this sequence the data from the samples that have a different behavioral characteristics or the different virulence, and then compare it with the reference genome that is characterized in the database. So let's these analyses and these different different technologies are made possible through the advances that takes millions and millions of cheaply generated short read sequences and align them to a reference that will help us extract meaningful insights in the biological system that we are considering. So this sequencing technology actually helps us to explore genetic variations like never before. Like we can use different types of this sequencing to address the particular specific need that we want to address. Like in the case of a targeted sequencing panel, we will have high coverage, but we are going to target only 40 to 400 genes in terms of human genome. Or if you want to target specific genes in terms of viral genome, we don't have to do that, because viral sequences or genomes tend to be very short in length, and people go for whole genome sequencing of pathogens, that is like virus, etc. But in terms of human genome, then we can use targeted sequencing or whole exome sequencing or whole genome sequencing to tailor the kind of sequencing experiment that we want to perform on the samples that we have. So these three different sequencing efforts, they have their own advantages and disadvantages. So some of the advantages that would categorize or differentiate them is the questions or the biological implications that they can be used to perform. Like whole genome sequencing can be helped, even though in spite of low coverage, can be performed on a large number of samples to identify specifically those variants which are very rare, and their association with the disease state or association with the phenotypical history that the samples are actually representing or depicting. Whole exome sequencing will give you a transcriptomic snapshot of the system at the particular time. So that is going to give you the expression profiles, or give you the expression quantification of all the coding genes at that particular snapshot. And targeted sequencing panel will help you specifically target little portions of the genome, so that we can perform, or we can understand their variation with respect to the reference sequence at a very high coverage, so that very rare mutations and very rare variants can be analyzed and identified and then addressed with a lot of confidence. So so how this is done? So this is done by by first collecting, or first sharing the genomic DNA into multiple small fragments of 200 to 300 base pairs, and then we attach the adapters to these fragments, and then these adapters are then attached adapters are then applied to the flow cell, where they are then amplified using a bridge PCR technique, and then these amplified shared fragments are then sequenced by the sequencer machine and these sequenced fragments are called as sequence calls depending upon their specific nucleotide in A, T, G, or C. So the NGS sequencing machine or sequencing technology basically converts, or basically takes the biological sample and converts them into a digital sequencing data that it gives us an output in the form of FASTQ files. So these outputs that we have from the NGS sequencing machines and NGS sequencing technology, or high throughput sequencing technology, are given us in FASTQ format. So FASTQ format is, yes, a FASTQ format, and we also, there is also another format that is related to FASTQ and is very very important in the analysis pipeline of this NGS sequencing data. That is FASTA format and GTA format. Let's go through FASTQ format first, and then let's come back to the FASTA format. So the FASTQ format, the FASTQ file reads like this. So the FASTQ file has a starting header which contains the important information such as the read name, the length, and different sample characteristics, and all the different characteristics that one can associate with this particular read. And the next line describes, the next line actually depicts the actual calls of the nucleotide themselves. So these are the actual reads, sequences that are sequenced by the NGS sequencers. And then we have a separator that is usually separates these base calls from the quality scores that is associated with each one of these base calls, and that separator is a plus sign. And sometimes this separator line is also populated with the same information that one finds with the header. So, and then after this separator, we get the quality scores that is associated with each and every one of these base calls in ASCII format. So these, each and every one of these characters represent, or they actually tell us how confident we are, or how confident the sequencing technology is about the placement, or is about the call of this particular particular base at that specific position. This is used right throughout our downstream analysis, where first this is used to filter those reads which are of low quality, and then even until the end, where we can associate our variant through the variant identified variants with the quality score that that it is attached to, or that it is called with. So let's go back to the FASTA file format. This is another file format which is usually used in the NGS sequencing pipeline, where when we have to map the FASTQ files or the reads in the FASTQ files format to the reference genome. So FASTA file format is usually reserved for the reference genome, where this file format is a very simple text-based file format, where the first line usually starts with a greater than symbol, and then what follows is the header for this FASTA file for the whole genome sequence of this file. So this header will have important information like accession ID, the name of the species, and in addition to that, they can also have the strain names or isolate names, or the type of technology used to sequence this data, and etc. etc. And after that, what follows is the actual whole genome sequence of this particular species. For example, in this, it's Spodoptera, and in this case, it's a rabies virus. So this is the genome sequence right from the first nucleotide to the last nucleotide given in a one single line or one single entry. So this doesn't actually describe in terms of eukaryotic genome which portions of this genome sequence are coding sequences, or which portions of this genome sequence are non-coding sequences, or which portions are exon or intron in terms of within the gene. So that information is actually loaded, or that information is actually supplied with the GTF file. So or GFF file, which tells us at what position can we find the coding region, at what position can we find the exons, and what position can we find the non-coding regions of this in this FASTA file for the reference sequence. So these three are absolutely essential for performing a NGS pipeline analysis of sequenced data. And what happens is the FASTQ files, which represent the reads that are sequenced, they are mapped onto the reference genome, which in the FASTA files. Here you can see the reference genome, and then these are the reads that are mapped onto this particular position, specific position, based on the sequence alignment, or based on the sequence identity on this one. So here, this is a detailed view of how this is performed and how this can be visualized. So in this representation here, this gray bar represents the genome sequence, and this big gray bar represents the gene, and these ones are non-coding areas, non-coding regions, and these blue bars and red bars represents the red bars and blue bars represents the forward strand and reverse strand respectively. And they can be mapped like this onto the reference genome, and then identify, and then their specific and the variation that is present in these FASTQ files with respect to the reference genome can be identified, and then they can be characterized. So to visualize the whole mapping and the whole and the processes of this mapping and output, they can be visualized using some visualizing tool like JBrowser or IGV, and these are the standard visualizing tools that one uses to visualize this mapping algorithm. So here we have the reference sequence that is mentioned in this one, and then what follows is the consensus, or what follows is the coverage of this mapped reads. So we do not have a uniform coverage because of the technology. It is not uniformly read, or it is not uniformly sequenced at all the levels. So these chunks of small FASTQ files, as these chunks of small reads, they get mapped to the genome, and based on their based on the coverage. Like here we have really high coverage where we have very many reads that are aligned at this position. So we see that the coverage is really high. But here the coverage is very low or very shallow because we do not have a lot of reads at this particular position. So these can be visualized, and in addition to visualizing the mapping of these reads onto the genome, we can also identify specific mutation at these positions, which can also be visualized in this one. So let me quickly go to Talbot Bioinfo platform and show you how to run a simple visualization pipeline for a viral genome. So let's take it forward there and then come back again to here. Share screen. Yeah, can you see my screen? I think you can see my screen. Let's go to server.tbioinfo, and then here, if you go to genomics analysis, and then under mutation variant and parallel analysis of mutation variant data, you drop down. So we have prepared some demo pipelines and demo pipelines, which will give you the feeling of running this analysis or running this pipeline, building up this pipeline for this sequenced analysis. And then you can access the output files, and you can perform the similar analysis that we are performing in the using the output file. You can visualize, you can perform downstream analysis with the output file, and you can learn what these files can be, what these pipelines can be built for, and how the output can be leveraged to perform multiple analysis. So let's build this start. Here usually the input files are uploaded because it is a demo pipeline, and we then after starting we map the reads onto the genome using Bowtie2 algorithm, and we go there, and then after this we complete this pipeline with visualization tool, and we end the pipeline. And after you end the pipeline, you get access to download all the pipeline results, where we have the mapping statistics of these reads mapped onto the genome, and we have the sequence alignment map or SAM file, which is the alignment file, mapped file in a SAM format. This can also be given in a binary alignment mapped file in binary format. And we have the input FASTQ files that we have provided for this mapping or provided for this analysis. And this is the, I think this is the reference genome. And then if under this table, under this section, there is a small clickable link called visualization. If you click this, okay. If you click this visualization, usually this should give you a visualization like this. Let's try for some other format. And yeah, I don't know why this one is not working. Okay. So you follow the same process, you map it onto the reference genome, and you visualize. Okay, some issue with this, which will be sorted out later. Let's go back to the PowerPoint. So I can show you in the PowerPoint how you can visualize this. So this is what the visualization is will look like, where you have a lot of reads attached or aligned to the reference genome, and this will tell you, or this will characterize the sequence coverage at each and every position of this reference in this reference genome file, and also it will also characterize, so it will show you the variants that you can identify, or the variants that you can observe in the sequenced reads that you have so far. So let's move forward and see. This is just one set of analysis where you can leverage the NGS sequencing um, technology to perform sequencing on the sample of your own. But you can also, in the event of available sequenced data, you can also perform a comparative analysis, which will give you a comparative analysis where you can collect different whole genome sequences from the publicly available repositories like NCBI virus, and then you can perform a multiple sequence alignment and a comparative analysis of these whole genome sequences of different viral strains that we can collect. For example, here we can collect all beta coronavirus genomes, and that are characterized by different countries, from different hosts and different countries of origin and different strains, and etc. etc. So we can perform a multiple sequence alignment of the whole genome of these different multiple strains of this SARS virus, for example, and that is going to help us identify those specific variants, or those variants that are specific to these strains or specific to different location of isolation, location that these viral samples are obtained from. And this multiple sequence alignment that we will perform on this whole genome sequence data will form the bedrock, or will form the basis on which a phylogenetic reconstruction can be done, or can be performed using the pipeline again. And this will actually give us those sequences or those strains that are closely related to each other in terms of evolutionary, in terms of evolution of these viral strains, and they are not closely related, or they are far away in terms of their viral evolution. So there are multiple algorithms, or the multiple phylogenetic reconstruction method that is available in the pipeline to perform such an analysis, and this analysis will help us identify which sequences or which strains in this case are close together, and we can relate their variants to their grouping by, for example, in this case, strains, or we can also find out whether these different groupings that identified by the phylogenetic tree can represent their other phenotypical characteristics, like different geolocations, or different variants like Delta and other Omicron variants, etc., etc., that we can use it for. So we can extend this multiple sequence alignment into analysis of viral proteins, where in addition to identifying variants in the nucleotide sequences, we can identify, or we can extend that to the amino acid sequences that these genes code for, and that will give us the mutations or variants, or that are specific, or that are that we can relate to the viral proteins. For example, this can be leveraged to do that. And in viral proteins, we will focus today on the glycoprotein or spike glycoprotein, where one can extend this mutation and extend this multiple sequence alignment to glycoprotein, for example, and identify differences in the glycoproteins or surface protein, spike protein, between different strains. That will help us relate to the virulence or the pathogenicity of this particular virus when this virus recognizes the host cell using this spike protein, or using this glycoprotein, using other different proteins that the virus deploys. So this analysis will form the bedrock, or will form the basis of vaccine design as well. So if one identifies from these multiple sequence alignment those stretch of sequences that are common. Let me go back. Okay, it's taking so long. It's not responding also. Yeah. For example, in this case, if one identifies those stretch of sequences between different strains that are common and those stretch of sequences that have higher tendency to get mutated or higher variants, then we can specifically design our vaccines to target the common regions of these surface proteins. So that becomes one of the strategies in which these vaccines are developed, and that's how these vaccines are targeted and and they can, when targeted and when these vaccines are deployed, they can interfere with the ability of these surface proteins to bind to the host cell and then impair their virulence and then help us in developing necessary immune response to these particular ones. So this is how this is actually coronavirus vaccine is targeted using one of the targets is a spike protein, and then we can also using similar analysis identify stretches and identify those regions where the antiviral drugs can target, and these drugs can also be helped to bind to those regions of these of different proteins in the virus or different proteins that are specific to the virus, and these proteins are essential for the virus to survive as it involves in the necessary viral propagation of system. And if we can find specific inhibitor that will stop or that will affect or that will impair the function of these viral protein in specific, then this can also be leveraged. So the comparative sequence analysis of whole genome sequences and then extended to comparative analysis on the protein sequences that these genes encode specifically from different strains of virus and different from these different regions these viruses are isolated from, will form the bedrock of such biological advances that we are getting, like the vaccines and like other interferences that we can create to stop the pathogenicity of this virus. So we have prepared such a project in this in the Omics Logic platform, and this will you will have a lot of information, and you'll have a detailed lessons on how to perform these analysis and how to use the Tobot Bioinfo platform to perform these analysis, so that related biological inferences can be implications can be performed and can be related to the pathogenicity or to the virulence of this virus or different infectious agents. So that will conclude my session today. I will hand it over back to Sparsh. I think Sparsh is going to take over from me.
Thank you so much, Dr. Raghavendran, for such an insightful session. So let me just share my screen.
Hi, Sparsh. This is Mohit. Actually, before you start your presentation, I also wanted to show like everyone a little bit about the next session, what it will be about. And also we have a wonderful opportunity with another workshop coming up on SARS-CoV-2 variant analysis. So I wanted to showcase that as well. So can I do
Sure.
that? And then quickly you can.
Sure,
All
sir.
right. Thank you. Is my screen visible, Sparsh?
Yes, sir, your screen is visible.
All right, so the next session will be about on Friday, February 18th. And we will start a little bit early for this session, the first session then. Basically, what we are going to do is to understand the NGS workflow. So before starting the NGS next generation sequencing workflow, usually what we do is isolate and purify your nucleic acid, and then DNA extraction methods also can introduce inhibitors which can affect the workflow. So there are some other steps that we are going to discuss, that what is being used before data analysis. So, I mean, we will also discuss that different methods for virus discovery, right? Like cell culture inoculation, sequence independent PCRs, they have been used. And now the emergence of NGS that has entirely changed the field of virus discovery. So we discussed that in the first half of the session. So in the next session, we will be discussing about this technology, Illumina technology. So, so Raja Varman Kittu, a clinical genomics specialist from Premas Life Sciences, a company which is focused on Illumina sequencing and is specializes on helping scientific community with the instruments and the analysis and all of that. So so Raja Varman will be letting us know, will be helping us learn about all of that, the initial steps. So, and it would be followed by another session, I mean, by one of the experts from Premas, which will be about finding NGS data from public repositories. So we will be looking at, once the data is prepared and it is being generated by the machine, how do you go to the next step? How do you prepare your data to be submitted? And when it is submitted and it is on a public portal and it is on a public repository, how do you understand where it is? How do you find that information relevant to your project? And also getting to that data and how to interpret that entry. So that is one of the challenge that many students face. So we thought that it would be nice to have a guest session in this program where experts who are working on this every day can come and tell you about these things, and so that we can all learn about it. So that's about the two sessions that we have next on February 18th. But we have a lot of upcoming wonderful opportunities and exciting sessions coming up in March. So in March, we are going to start with a program which is a short workshop for 3 days. It's about genomic surveillance of SARS-CoV-2 variant of concerns. So in this program, we will be speaking about importance of genomic surveillance for novel emerging variants and consequences of large-scale vaccination on its evolution. So as we were discussing that throughout in this presentation. So in this short workshop, we will be doing different kind of activities. So that would also include like generating a report. So if anyone who's working on coronavirus, SARS-CoV-2 and other viruses, and if what is the purpose of doing that analysis? So generating and finding that in a form of a report that actually helps you understand the analysis in a very simple way, and also to do this at a genomic level. So this program will be like this short workshop will cover all those things. So let me quickly take you through the sessions that we have planned for this short workshop, starting from 1st March to 3rd March. So we will start with a session that would be about NGS, RT-PCR, and how to detect viral particles, sequencing of genomes, and detection of variants. So we will be talking about established protocols like Illumina RTIC protocol used for full genome sequencing and PCR primers for variant tracking. So this would be the first introductory session that is for genomic surveillance, where we will be learning about all the data collection methods. Then in the next session, we will be learning about using NGS data to find using one second, please. So yeah, in the next session, we will be reviewing, it will be an hands-on session completely. So here we will be learn to load files from NGS pipeline into R using custom scripts, and how this is, and then before that, we will be learning how to build a pipeline and how to understand the outputs of these different algorithms that are used in a workflow, which is a pipeline for genomic analysis or viral genome analysis. So this will be the second session that will be an hour after the first session. And after that, we will have another introductory session on nature of emerging variants, why they are causing concern, what might be the driving force behind the observed clades or substrings of this virus as it continues to spread around the world. So that's the overview. And in this session, we will be learning about from a data perspective. So we will be going to NCBI virus, and we will be looking at different strains, and then we will be doing analysis on the Bioinfo platform, where we will be learning about how they differ and how they're emerging and connecting it to the phylogeny. So this is the first session, which is another beginner's day. These are the first day plan. It will be for 3 hours or 2.5 hours. And the second day, which is the day 2, which is on Wednesday, 2nd March, we will have an asynchronous session, which would be not a live session. In this session, we will be expecting the participants who are joining the workshop and doing it with us to learn about these different resources and courses that you can already do it in the infectious disease program as well. But in addition to that, there will be assignments to some COVID related research and analysis. So this is the day 2, and after that, the third day would be a research level meetings. We will have research level analysis. So in this session, what we are going to do is that we are going to learn about. One second, there are some chats that we are getting. So in this session, we are going to learn about the hands-on analysis for the logic behind like developing a workflow. So what are the different variant workflows that we are using, and how these samples that are varying from like clinical samples to environmental samples to wastewater, how they are being utilized to do this sort of analysis, and how many universities and many PIs and a lot of data is emerging from these sort of analysis, and what is the next step and how it is going to help us. So all of that will be covered in the introductory session, followed by the research level session. That would be followed by the second session, which would be about the downstream analysis of genomic data, evolutionary studies, and then phylogeny and origin tracing and reporting. So this is another specialized session, designed for those who are who have interest in going in depth into the variation, understanding understanding how it could impact at a level of the research question. So this will be the next session. Followed by that, we will be learning how all of this could be made into a report that could give us insights about this entire analysis that we have performed for variant, and how that correlates for vaccine implications, or as a structure level, how it reflects. So understanding the impact of the mutation and finding from the heap of data and like all those difficulties that make this analysis really challenging, we would be simplifying that throughout this entire workshop. And one of the reasons what we are doing this is also that we are launching this as a research support. So many who are interested in analyzing their data, we will be able to help them analyze their data through this. So this is what I wanted to share, and I would be looking at the questions if there are any, and I would really love to help those who have any queries about this. And now I'll pass it back to Sparsh to take this forward. Thank you, everyone.
Thank you so much, Dr. Mohit. Let me just share my screen. Dr. Mohit, could you please confirm if my screen is visible or not?
Yes, but your screen is visible. And once you're done, please hand it over to Dr. Raghav. So he wanted to show the visualization part. It's working now.
Sure, sir. I'll do so.
Okay, perfect. Thank you.
So hello, everyone. Hello, everyone. I'm Sparsh, and I'm going to quickly guide you through with the program page of Bioinformatics for infectious diseases. So this program is an online 1-month mentor-guided program and will end on 21st of March. So I'll just share the link of this program in the chat box. Just a moment. Here's the link. Yes. So in this program page, you will go through with the program overview. And here is the syllabus of all the interactive sessions which will be there, which will be included in the 1-month mentor guided program till 21st of March. Now, those who haven't enrolled themselves yet for the program, I am briefly letting them know about the 3 level of subscription uh, they need to choose from. So the 3 level of subscription, one of the level of subscription is beginner level of subscription, and the fees for that level of subscription is $150. And the resources which are included under that level is online tutorials, project examples, cloud pipelines, coding sessions, session recordings, live group sessions, and certificate for completion. Now moving on to the second level of subscription, that is intermediate level of subscription, and the fees for that is $250. And the resources which we'll be providing is online tutorials, project examples, cloud pipelines, coding session recordings, live group sessions, plus one-on-one mentor support and certificate for completion. The third level of subscription is the advanced level of subscription, and the fees for that level of subscription is $350. And the resources include are online tutorials, project examples, cloud pipelines, coding sessions, session recordings, live group sessions, one-on-one mentor support, and you have an opportunity to work on your research proposal and present to the mentor for the evaluation. Then certificate of completion and also certificate of excellence. Now, we are right now providing with a good scholarship those students who are interested, or those participants, those who are interested for a scholarship can fill out this scholarship form. I have shared the link in the chat box, and after reviewing the form, we can get back to you. So is there any question regarding the enrollment process? If yes, so please put that query in your chat box. Also, for any query, we are available at marketing@omicslogic.com. The link, the email ID is in the chat box. And for Indian contacts, we specifically have a contact number that is 9876134120. So anyone is having any kind of query can mail us at marketing@omicslogic.com and can call us at 9876134120, specifically for Indian contacts. Now, I would like to pass on the stage to Dr. Raghavendran so as to continue with his visualization process. Thank you, everyone.
Thank you very much, Sparsh. Okay, let's say, yeah, it is good news because the visualization is back on, it is working. So let me show you how you can build a demo pipeline and then access the output file so that you can get a sense of what these analyses can give you and how these analyses can be performed and what all can you infer from these analysis. So let's quickly go to, let's quickly share screen. I'll share everything. I think that is going to be helpful. Yes, I'll share everything. And okay. See my screen, Sparsh or Mohit, somebody? Okay.
Yes, so we can see.
So
your
let's
screen.
go to the server. Thank you very much. So we go to the server Bioinfo platform, and then under genomics analysis, we go to mutation variant and parallel analysis of mutation variant data. And then, sorry, we have built these demo pipeline for students like you who are enrolled in the program to access these outputs and to access these pipeline building experience so that you can get a sense of how to work with the platform and what are the output files and what are the biological implications that we can get from the output file. So let's click the demo SARS-CoV-2 visualization platform. So this follows the analysis. We're not going to do mutation variant parallel analysis of mutation variant, but we are going to do, we are considering Let's say that if we have sequenced a new strain of SARS-CoV-2, or we have sequenced a sample of SARS-CoV-2 isolated from a patient we encounter. So we want to map it onto the reference genome and see what are the differences or variants that our particular sample contains or has with respect to the reference genome that we can map it onto, a reference genome that we can identify to. So in order to do that, we obviously have to start, and then after clicking start, the pipeline is going to give you all possible routes that you can navigate to. So the only route that we have right now is using Bioinformatics, because the input is it's a list of FASTQ sequences, and this is also here to simplify for users like you, so that you don't get confused with very many different options that are available at different points. So when you are going to build your own pipeline, and then by inputting the relevant input files in FASTQ format, then you're going to have options for pre-processing, mapping, and all the other options available to you. So you have to build a pipeline based on how you have understood the lessons, that the associated lessons, in which we are going to explain the logic behind this NGS sequencing analysis, and how to perform this in the platform. So let's click this mapping. It's going to tell you what it's going to do, and some associated parameters that you have to set. Most of the time, these parameters will remain in the default option, and if not, then you will have specific instructions on what to enter at which place in the associated lessons. So we now visualize the mapped sequences or mapped reads onto the genome using visualization, and then after this we will end the pipeline. So if you end the pipeline, now, as I mentioned, you get access to these files. You can download these files and visualize in your own computer, but that is going to take a long time, because this is like almost like 0.8 GB, these files. So let's click visualization here, which is going to take us to the inbuilt JBrowse tool, which will help us visualize this this analysis. So we let it load, then quickly check if any specific chat is there. Okay, we quickly, still loading. So first you are going to have not all of these ticked on, and yeah, so we will have our genome genes, and then we'll have reference sequences. So that is going to be populated here, and then we'll have the alignment, coverage, and SNPs here. Usually this is going to be only alignments. So you'll have not like this. Since I have used this, it's going to be like that. So let's if you see something like this, then I think we have to load it again. Okay, it says we have too much data to show. Let's zoom in. So let's zoom in. Okay, now you can see the reads that are aligned to the reference sequences. The reference sequence now you cannot see because you have to zoom in. Let's zoom in a little bit more so that we can see the reference sequences. And there you go. You can see badly. Yeah. So this is going to be your forward sequence. This is going to be your complementary sequence. You have all the 3 different amino acid translations based on the starting codon points, and we don't want those. We will actually turn those off for now, show translation, so that it simplifies our view. So that is good. So what you see here is actually barcodes, actually, because these are very closely packed. You can see like a band. So this is a coverage of this represents the coverage of our sequencing effort at this particular specific position. So you can obviously see at some portions we have some differences, or we have some minute deviations. So for example, this read, the red one, which is a forward strand, has a variant here with respect to the original sequence there. So we can see whether, if you double click this, if you click this once, then it is going to give you a window view of that particular read, the read that you actually clicked, and then that is going to give you the quality score associated with this particular position. We can actually check whether what is the quality score of this one. So I think this is not very good quality. How to identify this? I think yeah, that is why it is not called in, and also it is only present in only one of these reads, and all the other reads actually coincide or actually agree with the this particular position. So if you click this bar, this actually bar, we have 446 reads mapped onto this specific position, out of which 444 agree with the reference sequence, which is T or T in the forward sequence. You can see it's T in the forward strand, and only 2 of them are mutated variant to A. So this doesn't qualify as a significantly identifiable variant. So that is why we do not have any other marking in this particular position, because they occur only at two of our FASTQ files or 2 of our reads compared to the 400, 500 different reads. So that is almost nothing. So it's not statistically significant. So it is not called. So when you scan through the genome, you will find landmark areas like this. So here you will see that you have a marker. So if you click that, here you can see that there is a variant in this specific position, and this specific position has a very good coverage as it is depicted by this particular bar which goes from here. And if you scan anywhere in this identified position which qualifies this this variant as a significantly occurring variant, you can see that the reference is cytosine, and we do not have any cytosine at all. We have 442 of our reads mutated or having a significant variant to thiamine, and 2 of them to guanine. So this is how you can identify specific variants, and this is how you can visualize specific variants. This will be extremely helpful once you perform the statistical validation of these variants, and then we can come back to the visualization to identify in which portion of the gene or in specifically which portion of the genome does this variant occur, whether you have and how does other FASTQ files agree with that or not. So this is how we can do. So you can actually zoom out to navigate through this space in a much quicker way, and then when you do that, you can also appreciate the variations that, or appreciate the differences in the coverage that our sequencing effort has actually produced. So these are the regions where you cannot believe a lot, because you do not have very many coverage in these regions, but wherever you have bars really high, and that those places you can believe them. So this is just a very quick sample of how you can visualize the mapping of FASTQ files onto the reference genome. And and of course you do not have to go through, okay, well, here we have identified one more of these, and this is really highly variant. Did you see that? So you have a lot of variation in this particular specific position, and we have a deletion mutation also. You can see it's, yeah, so there is a deletion of one nucleotide here also. So that you can identify a lot of variants with this. So this is how you will visualize the variants. So while you go through the program, you will also have very how you visualize, how you can perform this analysis, and how you can visualize this, and how you can build a pipeline by yourself, so that you can perform sequencing analysis or build a pipeline like this to visualize your data. So if you have any queries, please let me know. I will take those questions. And if not, then I'll hand it over back to Sparsh. And thank you very much for your participation. Sparsh, yes, thank you.
Thank you so much, Raghavendran sir. So yeah, so I would like to tell you one thing. Those participants who are interested in scholarships, please provide us with your email ID here in the chat box. So we'll mail you about the scholarships. And also please add marketing@omicslogic.com in your mailing list, because sometimes spam gets triggered of bulk mailing. So do check mails in your spam folder and promotional tab. Thank you. Also, if we have some queries related to the session or the enrollment process, please feel free to put your query in the chat box so that we can answer those. Thank you, Sarah. Thank you, David, for updating your email ID. So I'm noting down the email IDs and we'll mail you out in just a short while. Do we have any queries regarding session? If yes, so please feel free to drop the question in the chat box. Dr. Raghavendran, I think so we have a question over here. So Prachi is asking, how do we select cut-off values for selecting the reads?
Okay, Prachi, if you can hear me, yes, I got myself unmuted. Okay, this is done by the FastQ quality control algorithm, which is part of the Trimatic algorithm that you will select when you are selecting your pipeline, and then you'll have a visual feedback of how these FastQ files how the quality scores of these FastQ files vary at each and every different positions and their distribution across different FastQ files. You can perform this FastQ quality control as a standalone package also in the utilities, and there by identifying which portions of your FastQ files or of your read files are dipping below the threshold, which is usually around if you go to if you use the FastQC HTML output, then it is given green, red, and yellow. So the top 80% or above, below, I mean, whatever scores that are above 80% are considered as excellent read quality. And what that comes around, around 30. Thank you very much. I mean, around 30. Yeah. So whatever goes down below that will be left for your discretion to identify or to trim it from the read file. So you can run this analysis as a standalone version and then decide how to or what are the different trimming that you can perform so that you can maintain an excellent FastQ files or read files. Yeah. In browse, some of the lines were purple and usually correspond to very low coverage reads. Are these variant sequences weren't that similar to others? I mean, you will find there are ways in which okay, I think she muted me also. Some of these in the JBrowse, only those that are that qualify as a significant variant, they are given those mutation landmarks. So others are just left like that, and you can actually turn off the other ones. These could be due to technical error. This could be due to low quality of that in that specific position. So you can go through those FastQ files to verify what is the issue there. If we have I have okay, and then sir, JBrowse are working like MegaX. I have no experience with MegaX, Naveen. JBrowse and IGV will have very similar display, but JBrowse obviously you can work it as a, you can use it as a we use it as a tool to display or help you visualize the heavy SAM file and BAM files. Yes, for visualization only. Yes, it is for visualization only. Yeah. And, but this becomes extremely important also, and you will have other statistical tools to validate your variants, like different variant calling algorithms that we will employ to identify mutations that are significant and statistically significant. So you don't have to visually go through each one of them and then note down the variant. So this is for visualization purpose, where you can relate to those mutants that we have identified in the sequence where they occur and how they occur. And in terms of viral genome, where you do not have a lot of genes and large number of sequences, then you can actually go through this visualization and identify how these relate to different structural forms of their protein that they encode. And I think that will answer some of the questions that you have thrown up. And then we have a question regarding the recording of this session. I think Sparsh can answer that, and when and how that will be made accessible. Yes. And any other queries related to the session today?
I think so, Dr. Raghuvinder. Prachi has a query. Her hand is raised. Prachi,
sorry, I did not know. Prachi, can you please? Sorry, I'm so sorry, Prachi. I did not notice. I think Prachi is not here. I think that's okay. So any other queries?
So about the session recording, yes, you will get a mail about this insights of this session surely by tomorrow itself. So any other queries regarding session, please?
Okay, thank you, Prachi. Thank you for letting us know. Taha, Baksh Taha, I think he has his question.
Assalamu alaikum.
Hello, Bakr. Yeah.
My name is Bakr Ahmad Taha. I'm PhD student in UKM in Malaysia. My project about COVID-19. I use biosensor laser for detection coronavirus on the service. Therefore, the bioinformatic information, can I get how extraction feature such as spike and envelope diameter?
Sorry,
can I
think can you use?
Yeah, can I get extraction feature for virus such as spike length and envelope diameter for the virus?
You can. I think
because I use image processing with the laser.
I get it. I understand your query. That's very interesting. I mean, in a way, Dr. Mohit, if he's here, I think he's not here, but in a way, you can get spike length from the protein itself, from the three-dimensional structure of the protein. Indirectly, you can calculate the ellipsoidal radius of that particular globular protein that will correspond to the spike length, but not in the way that you would identify. I mean, in order to get the envelope radius, you have to use some other data. You cannot use sequencing data to get envelope radius. No, I don't think it is going to be very related to envelope radius. I have no experience in this, and I find it really difficult to extract the envelope radius information from the whole genome sequence or even annotated.
Because this feature for detection by laser very interesting. Because I need the laser is blind. When I scanning any service, cannot I have knowledge about this virus or bacteria. But I use this feature to get the laser knowing this viruses by this feature, spike and the outline shape viruses. This I need use image processing to extraction this feature.
It's not allowed by.
Yeah, I think I don't know. Maybe by laser method itself, I think you can get the features as you were actually as you were actually explaining. But I don't know whether NGS technology can address your query in this particular regard. I don't think it can also do that.
Okay, thank you.
Thank you for the insights. Thank you. And any other queries? Okay, I don't see any. And then thank you very much for your participation one more time, and hope to see you all in the program, in the next session and in the program. Thank you very much. If nothing else to add, then we can hand it over to Sparsh if she has anything else to add to this.
No, it's everything is, I think, so clear. So, and maximum of the doubts are also clear, Dr. Raghavendran. It was glad to have you all over here, and looking forward to meet you all in the next session in the program itself. So, thank you.
Thank you. We also look forward to seeing you. Thank you.